In sports science, outcomes rarely depend on a single factor. If we want to understand what drives an athlete's explosive jump power, we must look at how categorical variables, like their gender and the specific type of nutritional supplement they use, interact at the same time. To see how this works in practice, we are going to analyze a sports science dataset. Our continuous outcome is explosive jump power, and we are testing how it is influenced by two distinct factors: gender and a supplement factor split into three levels: a control group, a carbohydrate supplement, and a protein supplement. We are going to take this raw CSV data and walk step by step through JASP. By the end of this tutorial, you will know how to turn these spreadsheet rows into a fully formatted, publication-ready APA results paragraph. Knowing how to correctly calculate the interaction between multiple variables is exactly what separates basic data collection from defensible scientific evidence. This is the JASP ANOVA setup. Place your continuous variable, jump power, into the dependent variable box, and both categorical variables, gender and supplement, into fixed factors. Under Display Options, check Descriptive Statistics and Estimates of Effect Size. Make sure omega squared is selected. Before we interpret any results, we have to perform a safety check. Like all parametric tests, a mixed factor ANOVA relies on a series of mathematical assumptions that must be validated first. First, we check the homogeneity of variance using Levene's test. We are looking for no significant difference in variance within our dependent variable groups. Second, we check our normal distribution using a QQ plot of the residuals. You want to see the plotted data points closely hugging the diagonal line, which confirms the data is normally distributed and linear. If your data fails these assumption tests, or if the initial ANOVA reports no significant difference at all, you must stop immediately. Proceeding further would only lead to false conclusions. Now that our assumptions are cleared, we can configure our visual output. In the Descriptive Plots menu, assign your primary factor, supplement, to the horizontal axis, and place your secondary factor, gender, as separate lines. When we look at the main ANOVA table, the p-values indicate significant main effects for both variables. This means there is a statistically significant difference in jump power between genders, regardless of the supplement, and a significant difference between supplements, regardless of gender. But we also need to check for an interaction effect. An interaction occurs when the influence of one independent variable heavily alters how the other independent variable behaves. By checking the specific interaction row labeled Gender * Supplement in the data table, we see a highly significant p-value of less than 0.001, paired with a medium to large effect size. This graph plots the descriptive statistics, showing jump power on the vertical axis against supplement groups on the horizontal. Notice how the lines for male and female athletes intersect slightly between control and carbohydrate groups, but sharply diverge at the protein mark. That visual divergence is the interaction in action. It proves that the protein supplement's effectiveness is heavily dependent on the athlete's gender. Because our overall ANOVA is significant, we can now run post-hoc testing to locate the exact source of these differences. We will apply the Tukey correction specifically to the supplement variable. We skip post-hoc testing for the gender variable. Since gender only has two levels in this dataset, any significant main effect already tells us exactly where the difference lies, making further breakdown redundant. Here is the resulting post-hoc comparisons table. Looking at the adjusted p-values in the final column, we see highly significant differences when comparing the control group and the carbohydrate group against the protein group. However, there is no significant difference between control and carbohydrate. As an alternative to a blanket post-hoc test, researchers can use the Contrasts menu. By selecting Custom from the dropdown, you can manually input values to test specific, targeted combinations of cells based on your exact research question. We can also use Simple Main Effects. This feature performs limited pairwise comparisons. In our case, it allows us to isolate the data and evaluate the distinct jump power difference between males and females exclusively within that high-performing protein group. These tools, post-hoc tests and simple main effects, allow us to take a broad interaction finding and drill down into a specific, actionable insight that we can report with confidence. Now that our analysis is complete, we move from statistical computation to scientific reporting. Our final step is mapping these numbers into a formal written conclusion. We follow strict APA formatting for this. We take the F statistics, the degrees of freedom, and the main p-values directly from the initial JASP ANOVA output table and insert them sequentially into our text to state the main effects for gender and supplement. Next, we incorporate the secondary analytical data. We pull the omega squared values to report the effect sizes. Finally, we integrate the post-hoc p-values and the simple main effects findings into our concluding sentences to explain the specific interactions. Mastering this exact two-way ANOVA workflow allows you to process multiple variables seamlessly. You can move from raw categorical and continuous data to defensible, publication-ready scientific claims in mere minutes.